Hi, Assalamualaikum warahmatullahi wabarakatuh. Today our group 5 will present about dietary response of the breadfish Cladoceran Daphniosis australis feeds on different algae species.
This journal is about dietary response of Perkis cladoceran, which is Daphniosis australis fed on different algae species. The problem that lead to this research is because of low algae abundance in the environment, and then the size of algae in environment cannot be handled by animal filtration apparatus. The next problem is the alternative food, which is non-algae food, contain low nutritional value. In this research, Daphniosis australis was fed using two type of food, which is monoalgae and multialgae. Monoalgae, which is Tetrasalmis sucica, Nanochlorosis aculeata, and Isochrysis titian. For multi-algae, there's four type of multi-algae, which is the mixture of Tetrasalmis sucica and Nanochlorosis aculeata. Um, the mixed feeding of Isochrysis titian and Nanochlorosis aculeata. And then Isochrysis titian and Tetrasalmis sucica. The last is Tetrasalmis sucica and Nanochlorosis aculeata.
Next, there are 3-point culture of algae. First, the size of this algae was in the range 8 to 10 micrometer, 5 to 6 micrometer, and 3 to 4 micrometer for Tisucica, L. and N. aculeata respectively. Second, each algae species was cultured in 2-liter sterilized glass bottles provided with aeration under continuous light exposure. Third, this algae was cultured in a series of batch cultures with inoculations every 2 days to ensure adequate algae supply at the exponential growth pace over the experimental period. Then there are also have 3 points for stock culture of the Australis. First, a population of the Australis was established in 10-liter plastic containers in the animal house, Flinders University for over 100 genera. Second, females were originally collected from a saline lake in the Korong National Park, South Australia in 2005. Third, since then a laboratory population was maintained two, at 20 to 22 degrees Celsius and 22 to 23 PSU under 12, 12 hours dark and 12 hours light regime at 1800 LX.
About experimental design, a live table technique was used to evaluate the hypothesis. First, extracting a single pathogenic female from the stock culture and placing her in a jar with 50 ml media comparable to the stock culture. Then, when the first generation emerged, newborn units were collected and individually acclimatized in a jar holding 15 ml media from each machine generation. So, to minimize confusing result from that microalgae, the media was refreshed every day in a new jar. Temperature, salinity, and photoperiod were maintained. For the life history research, only third-generation animals were employed. The unit youngs, then 24 hours, were individually inoculate. The variables investigated is individual longevity, age at first reproduction, egg development time, total clutch, and totals of spearing. Next, each individual's growth was assessed on a daily basis because they enter part of the head to the end of the carapace. Statistical analysis is using the statistical software first data were examined before to analysis to ensure that the normalcy exception was met using skewness and kurtosis value. Then, homogeneity of fairness was determined following Sokal and Rolfe. So, one-way ANOVA is used to detect the treatment effect on each life history variable. And Turkey HSD test also is used for comparison between groups.
The sort of algae diets get a substantial effect on animal lifespan. Female T dissociate and live in the longest compared to those fed on other diets. Female fat and Jesuclid survive longer than all the fat-only Tahitian. For egg development, alga species have substantial impact on EDT. Female given an eucalypt Tahitian monospecies hatch their egg faster than some nourished multi-alga species. As first reproduction, alga species has a major impact on female development. For somatic growth, dietary types had a substantial influence on the uterus growth rate. So total egg clutch. The overall number of egg clutch produced female during lifespan was considerably influenced by algae species. Female fed on monoalgae, Suchicha has the most clutch, while female fed on mono Tahitian had the fewest. The number of egg clutch did not differ.
Next, for response to algae species at the population levels, first, net reproductive rate (RO). Feeding on different species algae has a significant impact on RO. For example, females had a higher RO when give T-Succica, but females had the lowest RO when give L. Tahitian. Second, generation time (GT). Feeding on different algae species affected the generation time of D. australis significantly. For example, females fed with L. tahitian had the shortest GT compared to others with algae combination. Third, intrinsic growth rate (R). Algae species combination significantly influence the intrinsic growth rate of D. australis. For example, when animals were given just L combined with N or T. succica, the highest R was observed.
Okay, next is the discussion. For the first point is longevity. The addition of different algae species can affect the influence of a single algae species on zooplankton, which is turn change the pattern of survival in cladosirens. Daphnosis australis live longer when fed the monoalgae Tetraselmis sucica than when given either Nanoclopsis aculata or Isochrysis tahitian. Secondly is development, growth and reproduction. The egg development rates in monoalgae species was faster than in multi-algae species. This means that nutritional need change between sexual maturation and egg development. The development and reproductive response of various grazing species are determined by their nutritional requirement. Specific dietary for various life activities such as development and reproduction.
Next is the impact of alga species on Diphylia australis at the population level. There are two approaches to achieve a high R value for a rapidly increasing population. First is animals' rapid maturity, and second is maximum reproductive production. Diphylia cuculata has the higher R value when it feeds as a coetus. Diphylia australis population growth has accelerated by feeding it either a monoalgae diet or multi-algae diet. Next is about monoalgae diet versus multi-algae diet. T. succica egg reproduction and lifespan, and for an oculata and Tahitian, a somatic growth and early maturation. Individual life history metrics favor monoalgae diets over multi-algae diet, but population growth parameter did not change when given either the monoalgae diet or the multi-algae diet.
In conclusion, the most effective food for Cladocerian that gives maximum population growth is Tetracermis sucica for monoalgae that support animal survival and reproduction. For multi-algae is Isochrysys and Nanochlorosis that reduce time to reach maturity due to high carbon content. I think that's all from our group. Thank you.